David Rednick of UC Riverside, studying the impact on the invertebrate average abundance induced by the introduction of guppies to stream systems. There are two fundamental kinds of biology in ecology, functional and evolutionary. The projects focus on evolutionary ecology, which itself takes two views. The first view is that ecology solely shapes evolution and that there is no interaction between the two because of differences in timescale. The second view, which is presented by Dr. David Pimentel, suggests that ecology and evolution do have a dynamic and ever-changing interaction with each other. One affects the other and vice versa. Where would be an ideal place to observe this interaction? The ecosystem of a stream is highly varied and has many factors that make it a good place to study evolution. Some examples would include current, temperature, and the amount of light that reaches the stream. Another factor would be predators, particularly invasive predators. For such an introduction, there will be, as Dr. Pimentel posited in several papers, population regulation and genetic feedback. In his words, density influences selection, selection influences genetic makeup, and in turn, genetic makeup influences density. While Pimentel's ideas certainly help set the foundation for this new form of evolutionary ecology, ego-ego interaction is a more general term to describe this interaction. First coined by Dr. Resnick, this interaction is one in which a species changes the ecosystem, which in turn affects the species selection and leads to species evolution as a whole. One of the project's goals is to show the beginnings of a potential ego-ego interaction, as well as observe the impact of invasive species in new environments. In this experiment, Tom Heatherly of the University of Nebraska and Dr. Resnick used guppies as the invasive species for 4 streams in Trinidad: Upper Wallaha, Lower Wallaha, Kaiwah, and Taylor. High predation guppies were used because studies have shown that they have developed a life history in which they mostly prey on invertebrates, the object of our study. This is a chart of Dr. Resnick that shows the different aspects of a stream ecosystem. In situ production means that that's parts of the ecosystem that were originally produced within that ecosystem, while loctiness production refers to the parts of the ecosystem that were originally produced elsewhere. From the diagram, you can see that any effect that the guppies will have on the invertebrates will have an indirect impact on the other parts of the stream's ecosystem as a whole. Thus, just with the guppies' introduction, the whole structure of the stream has the potential to be changed. Invertebrate samples were then taken regularly from control reaches, which had no guppies, and experimental reaches, in which a guppy population had been established. Previous results from the Lower Wallaha and Upper Wallaha streams, which had started the experiment a season earlier than the Taylor and Kaiwah streams, concluded that there was a depression on the invertebrate abundance as a whole. This project was looking to see if the results will now be reflected in the Taylor and Kaiwah streams, which would be proof of the start of a potential ego-ego interaction. After collection, the stream samples were then separated with coarse and fine seeds, which were then processed for all invertebrates. These invertebrates were then identified and measured. This data was then inputted into the computer where various statistical tests were ran. This is a graph showing the previous results from Heatherly and shows the overall abundance of invertebrates. The x-axis marks the years and the y-axis marks the invertebrate abundance. 2 results are of particular note. The lines with the dark colored shapes are streams where the canopy was intact, while the lines with the light colored shapes are streams where the canopy was thin. A thin canopy allows increased light to reach the stream and thus increase primary production, which leads to more invertebrates. In 2010, for both control and experimental reaches, you can see that the invertebrate abundance is higher in the streams with thin canopies. The second result is that in 2010, you can see that there is a significant depression in invertebrate abundance for the experimental reaches when compared to their respective controls. However, in 2008, you can see no such difference. The reason for this is seasonal differences between the years. 2008 was much wetter than 2010. The frequent flooding wiped out the invertebrate community in both reaches. As it takes around 60 days for the community to reestablish itself, no significant data was produced. 2010, on the other hand, was a very dry year, which allowed this difference between the control and experimental reaches to be seen. Therefore, there's the seasonality factor, which only allows the guppy's impact to be seen during the dry season. So although the months that I analyzed, February and April 2011, were during the dry season, they were extremely rainy months nonetheless. A table comparison between the average invertebrate abundance between the control and experimental ranges shows that abundance is low for both ranges and fails to show the difference in invertebrate abundance seen in Heatherley's results. In comparison with previous results, the highest number for invertebrates abundance here, 4,572, is among the lowest invertebrate abundance on Heatherley's graph here. What is really striking is that Upper Walhaha, the stream with a thin canopy, has a lower invertebrate abundance than Lower Walhaha, its intact canopy counterpart. Even the light effect, which was one of Heatherley's biggest results, was shown to have no impact here because of the rainy season. Therefore, our results do not show consistency of the guppy's impact per Heatherley's results. However, what does remain consistent between these results and the earlier findings is the very low average invertebrate abundance during the rainy season, showing there is a seasonality factor with this interaction between the guppies and their new ecosystem. So although the data shows that there is no significant effect, these results are only preliminary and do not include all the stream samples data. And since it is known that no effect is detected during rainy months, we cannot conclude that these results disprove the previous findings. So our next immediate step would be to integrate the rainfall data from 2011 and its distribution with our data to conclusively see if our results can be attributed to the rainy season. Then analysis of the entirety of the invertebrate samples would definitively show that the guppy's introduction does have an impact on the invertebrate community. I would like to thank and acknowledge Dr. Resnick for guiding me through this project, Yuri Reynoso for helping me organize the project's data, World Roberts for choosing me to help with this project, and Dr. Sean and CJ for accepting me into the marketing program.